All right, moving on now to kind of our last section really of the whole biology subject, where we're going to look at how biological knowledge is used in society. And we've kind of introduced this topic already by looking at how the technology works. And we just need to discuss a few things to do with specific examples of gene cloning and genetically modified organisms. And we particularly look at them in the context of social and ethical implications. So that is in the study design as something you need to be able to discuss. What are the social or ethical or biological benefits of something, and what are maybe some risks associated with it? And then in our next lesson, we're going to look at this kind of final thing, which is strategies that deal with the emergence of new diseases, which is obviously very, very relevant this year. So today we're just looking at these two. My video will be quite short today, so take your notes down from that. And then there's a 10-minute video I want you to watch with some questions connected to that, and then some past exam questions that show how these questions tend to come up in exams. So in the last lesson, we looked at using bacterial transformation as a way to clone genes, so to make lots of copies of a gene. So this process can be used to create copies of genes that can be extracted and studied. And this has been a huge development in terms of helping scientists understand genetic disorders. So things like different types of cancer, things like diabetes, even things like Down syndrome. So all of these things that have a genetic component can be studied because of things like gene cloning. We can also use this to create copies of healthy genes that can actually be inserted into patients with genetic diseases. That is quite a tricky thing to do, and it generally has to be done when a person is still an embryo. So it's quite difficult. It has to be picked up early, but that is something that can be done now, is giving someone copies of these healthy genes. We mentioned this already, that gene cloning can be used to mass produce specific proteins that can be used to treat diseases. So the great example of that there is insulin. So cloning the gene and then inducing the gene expression so that the protein is produced. Okay. There are some implications, or I guess considerations with this type of technology. If you want to pause and have a think for a second what some of the benefits and some of the risks might be, you can do that. But some of the benefits of using gene cloning technology is that we're able to treat diseases that we couldn't treat otherwise. And this has a huge impact on the amount of pressure and money that is spent by hospitals. Obviously, being able to treat diseases can save people's lives or improve the quality of their life. So letting them live kind of healthier lives. By using bacteria to clone genes or using technology, this often reduces the need to use animals for testing. So traditionally, a lot of medical and pharmaceutical testing is done on animals. But if we can start finding different ways of doing that, animals aren't needed either to study diseases or to produce proteins. So in the past, insulin would actually be taken from animals like pigs or sheep or cows before there was a way to manufacture it. Also, by improving people's lives and people living longer, that will increase the population, which you'll see kind of comes up on both sides. This can be a positive thing. It can benefit the economy because we've got more people working, paying taxes and spending money. That can be a positive thing for the economy. Some of the risks here is that anything that involves even selective breeding, but particularly things like genetic modification, is we're starting to interfere with genetic variation. So if we kind of, get rid of faulty genes, what we think are faulty genes, and replace them with what we know is a healthy gene, there might be other components of that gene that we're not aware of that are really important. And if you remember back to natural selection, if we go through our steps of natural selection, the first step is variation. We need high genetic variation for natural selection to continue to occur and to make sure that we always have, the fittest by having this variation. So there's the risk that by interfering with that, we're decreasing that variation. Anything that relies on technology and medicine and is generally quite expensive can lead to inequality, because often these things are only available to people who can afford to access this type of technology. So we might end up with, wealthy people who are able to get it, and therefore they live a really long, healthy life. But poorer people can't access things, and they're more likely to get sick. This is a really interesting one. A lot of people say that, we're kind of at the point of playing God. And obviously that's just a phrase. It doesn't matter whether you believe in God or not. But this idea that as humans, we're interfering with the natural course of life and the course of events too much, and we don't know what the future consequences of that can be. Okay, there might be things that we are messing with that we don't understand yet. This leads a bit to the inequality as well, but obviously at some point some scientists or some politicians will be deciding which diseases get studied and treated over other ones. So that can be a risk, that we're kind of deciding to prioritize certain diseases and therefore certain people over others. And then, like I said, with this, there's kind of a double-edged sword here, that by too many people surviving, we can get overpopulation. And this can be a huge burden on the economy. If we've got a really aging population, that costs a lot of money, but also overpopulation. Too many people, not enough jobs, not enough opportunities, not enough housing, things like that can be a real issue as well. Okay, so it's really important in biology that you can look at benefits and risks. You need to be able to see both sides of these kind of social, ethical, and even biological implications. Okay, we're going to now focus on this. This is, oh, sorry, I didn't mean to cross that out. This is a particular area where DNA technology is used in science and in particular agriculture. So genetically modified organisms, or GMOs, involve altering an organism's genome using technology. We'll come back to this in a second, but this is a big distinction. Okay, so it's actually changing the DNA of an organism. This could be an animal. More commonly, we see it in crops and plants, and that's really because of ethical reasons. But it can be done in animals. It could be done in humans. It's not currently being done too much because there's a lot of regulations. In general, this is done to give an organism desirable traits, and we'll look at some examples of that in a minute. It can involve inserting genes into the genome of an organism. It could involve removing undesired genes from an organism's genome. It can involve altering genes by replacing nucleotides, and you're probably, if you're looking at that, thinking, well, that almost sounds like a mutation. Well, it is. That would be kind of inducing a mutation, causing a mutation by changing a sequence of DNA. It's really important to note here that genetic modification is different to selective breeding or artificial selection, which we looked at when we looked at evolution. Selective breeding involves choosing two organisms that have desired traits forcing them to breed with each other so that the offspring have good traits. Okay, so two dogs that have the same snout that we want, we select them and cause them to breed, so therefore their offspring has it. Genetic modification is different because it involves changing DNA using technology. Okay, so if we looked at it in that example of the dogs, it would mean actually inserting the DNA for that particular snout shape into the embryo of a dog so it has it. Okay, they are very different. They can have similar results, but they're very different methods, and it's important you understand that. Okay, the study design also specifies that you know the difference between a GMO and what is called a TGO. And we'll go through this in a second. If DNA is inserted from another species, that becomes a transgenic organism, or a TGO. So all transgenic organisms are also genetically modified organisms. So a genetically modified organism is any organism that's had its DNA altered. So a TGO has had its DNA altered, so it's a GMO, and it's been altered by a different species, so therefore it's a TGO. It doesn't work back the other way. So you can get some GMOs that are altered with DNA from their own species, so therefore they are not TGOs. This might be a better way of looking at it. So GMOs is any organism that has had its DNA altered or modified. So it could have some DNA removed. It could have some nucleotides, just specific nucleotides removed. It could have DNA inserted from a member of the same species. And we call that cisgenesis. Cis meaning kind of the same, or as it were created. TGO are a subsection of GMOs where the DNA is inserted from another species, and we call that transgenesis. When you look at the questions I've given for today, this comes up in the exams quite a lot. It'll give you an example. Is this a TGO or not, and justify why. Okay, so if it comes from another species, it's a TGO. Remember, I think this is what trips people up quite a lot. Often, the genes that are inserted maybe come from a bacteria into a plant. And people often look at that and go, well, bacteria aren't, a plant, so therefore it's not TGO. Bacteria are living organisms. So if we take the genes from bacteria and put it into a plant, that is going from one species to another species. That would be a TGO. We'll look at some examples and hopefully we'll kind of clarify this a little bit. So GMOs are primarily used within agriculture, and there's a few kind of main benefits or uses of them. One thing that GMOs can be used for, so we can modify organisms to give the crops resistance to either insects or herbicides. Okay, so if we give resistance to insects, it means that insects don't eat the plants. Okay, so that's obviously a good thing for farmers because insects aren't coming along and eating their plants. If they have resistance to herbicides, herbicides are used to kill weeds. And so often farmers will spray their crop with herbicides to kill all of the weeds, but it's important that the crops don't get killed as well. So the crops have resistance to those herbicides. An example of this comes up quite a lot, this term BT corn, or there's other BT crops. BT crops are crops that have been given genes from a particular type of bacteria, which is actually the BT bacteria, which produces a protein that kills insects. Okay, so it's an insecticide. Okay, so this happens in corn, it can happen in eggplants, there's different types of BT crops. So those crops are given genes from bacteria. So it is a GMO, yes, it's been modified. It is also a TGO because the gene has come from a bacteria and been placed into a corn crop. Two different species. It's a transgenic organism. Another example is using GMOs to increase the nutritional value of foods. An example here is this stuff called golden rice, which is one on the right here. And this is rice that has been modified with genes from a type of daffodil plant and also a specific bacteria. So two genes here. And when these genes are combined, they produce a protein that helps the rice to produce vitamin A. Okay, so it's basically increasing the nutritional value of rice, and therefore it can be great in low-income countries where maybe there's not a lot of food security. Okay, so what about this one, TGO or not? So we've got a rice crop that has been given genes from daffodil, a flower species, and a bacteria. So yes, this is also a transgenic organism. It's the genes that have been given to it are from a different species or two different species. Final example is using genetic modification to delay the ripening of foods, which increases the shelf life. Okay, so tomatoes generally actually become ripe very quickly, and then obviously if something over ripens, it rots within 1 to 2 days naturally after being picked. But obviously that's not great for farmers, it's not good for supermarkets, it's not good for people because things go rotten very quickly. So an example of using GMOs for preventing this was a particular brand called the Flavor Saver tomato. This tomato had the gene that causes ripening removed so that the proteins that cause the fruit to ripen aren't produced, and therefore you could get maybe a week, even longer, before the fruit starts to rot. So in this one, we've just removed a bit of DNA from the original organism, but not replaced it with anything. So that would not be a TGO. It's a GMO, it's a genetically modified organism, but it hasn't been given DNA from a different species. It's just had some of its own DNA removed. So that is a GMO. Okay, we'll go through the social and ethical implications of genetic modification, and then I want you to watch a video on it. So some of the benefits, it can make more productive crops because crops aren't being killed by insects or weeds, so the crops are more likely to survive. It can decrease habitat loss. Okay, so it can actually be good for the environment in a few ways. Firstly, if it's more productive, we need less land, so you don't have to cater for farming not working. Also, if crops are resistant to insects, then farmers don't need to use insecticides as much, so the sprays that kill insects. It can improve nutritional value of food, which we talked about before, which can increase health. It can increase food security, so there's more food available around the world. It can be more efficient and therefore profitable for farmers. It gives better quality food for us as the consumers. Obviously, we want nice quality food that doesn't rot straight away. But there's also some risks. Same as with gene cloning and using those kind of genetic technologies, we can decrease genetic variation, which can be a big issue because we don't know what other genes exist in plants that might be really important if, say, a new virus comes along. They are expensive, and that cost carries through to farmers and then obviously through to us. This can then create inequality because only some farmers are able to afford genetically modified crops. It is often viewed as unnatural. Again, that whole kind of, playing God element. And finally, because plants cross-pollinate, if wind blows seeds from one farm to another, you can get kind of mixing of GMO and non-GMO crops. So if you've got On one side of the road, you've got a GMO farm. And then over on this side of the road, you've got, you know, a certified organic farm where they don't use any sprays or anything like that. And some wind comes and blows some of these crops over here, that's going to contaminate this organic farm. Okay, so that could be a big issue for the farmers on that side. Okay, so we've had a bit of a look at the social and ethical implications of gene cloning. And also we've looked at what genetically modified organisms are, what transgenic organisms are, and some of those social and ethical implications of using those technologies. So I want you to go into the Google Doc now. There's a link to a video, I think it goes for about 8 minutes, some specific questions related to that video, and then some past exam questions, including one, I think it was either last year or 2018, that was 8 marks just on genetic modification. So that's quite a big chunk of marks in one exam. So it's good to see how those questions get asked.